So let's start with module 1 for the 2021 paper. Alright, let's see what's happening here. Bioenergetics, lovely, the easiest module in the entire syllabus. Alright, so what we're looking at here now, an experiment to observe the interconversations of compounds found in the nitrogen cycle was carried out by a student. So 150 centimeter cubes of a solution of urea, right, was mixed with 50 grams of a well aerated loam soil, right? The ammonium, nitrate, nitrite, and nitrate concentrations were immediately determined semi-quantitatively, right, by the use of test strips that produce color changes, right? The strips were then compared to a specific color scale, which gave a measurement of the ion in parts per million, right? I think it would have been better to use UV spectroscopy. Okay, moving on. So the mixture was sampled, right, for both ions in 6 hours, right? So 6 hour intervals, right, for 24 hours. Okay? I can use strip to find out how much is in part per million. Them people are never the chemistry. That's crazy. All right, so we're looking at this graph here, right? How it changed in parts per million. We have the ammonium concentration, which are the rhombuses, and we have the triangles, which are the nitrite rather concentration. All right. Everybody understands the graph, right? The graph is easy to interpret, yes. All right. So looking at that, all right. Let's see what the question asks now. Wow, we're asked to construct a table from the data. So instead of them asking us to construct a graph from the table, right? We're asked to now construct the table from the graph. Are you guys able to do this? Would you guys be able to do this?
Yes, sir.
What would be the headings in the table?
Concentration of ammonium ions and nitric ions. The
Okay,
units being part.
and then period of incubation. Oh, yeah, the units in part per million, lovely. And then you're gonna have incubation in it as well
Yes, sir.
per hour. Okay. So how many columns they gonna have? What? Four or three? Three columns.
3.
You have the incubation, then you have your ammonium versus the nitrite, and okay. All right, lovely. So we have that there. Shouldn't be too much. So just draw a table there. How many sides does a table have? Not the silence. Everybody's doomed. How many sides does a table have?
4.
Lovely. All right. So the table has four sides. So the thing is, though, it's because some persons in the heat of things they tend to do their tables like this, and then leave like no bottom to the table, right? Tables have titles as well, right? Tables have titles. And we don't write units going down like parts per million, parts per million, parts per million. No, you just put parts per million in the entire title and put it in brackets. There are things that you guys generally know how to do, and this is what labs do, right? You do the labs to develop your scientific skills, right? So you guys should know how to construct a table. I'm sorry for insulting your intelligence. All right. So here now, we're asked to identify the processes here. What would be process A? Process A?
Decomposition.
Decomposition. Or what we love to call decay. All right, lovely, lovely, lovely. What is B?
Ammonification.
Why would you say such a thing?
So it's not ammonification.
Well, I didn't say it's not ammonification, but why would you say such a thing? What is ammonification
So
done by?
because
Oh, go ahead. Tell me why, and then tell me the organism that is doing it.
Azobacter. I don't know.
Okay, so since Mashim, alright, I will not continue going through what causes what, okay? Alright, that's fine. Since Mashim. Because the thing is, no, we're not saying it with the vigor, the alacrity that I want you guys to say it with. Alright, so
Buster, was I right on the
uh, okay, let's put it to a vote. How many persons think that Azobacter contributes to ammonification?
Azobacter,
Yeah.
isn't that for nitrogen fixation?
Okay, so what we're going to do, we're going to mute the mics. Right? So we're gonna mute the mic. Okay, the thing is no. Azobacter, right? Okay. What does Azobacter do? No, this is a serious
So what did
question.
you say?
Right, what does Azobacter do? No, you're right.
Um,
You're right, right? So you're right about the nitrogen fixation part, But like, I'm not sure if we do we remember what. Okay, so we have nitrogen fixation, right? It is done by either rhizobium in a symbiotic relationship, or it is done by Clostridium or Azobacter, right?
Oh,
So that is what it does. In ammonification, we have what causing it?
Is there Pseudomonas?
Pseudomonas, lovely, right? So I have Pseudomonas and other types of bacteria and fungi, right? For C, C's what here? We have a ammonia nitrate,
Sorry, the
nitrification.
and the first step of nitrification is done by who? Ammonia nitrite,
Nitrosomonas. Is that the name?
nitrosomonas, right?
Yeah.
So I'm gonna add another one as well because some persons love the increase in information. So it's either done by nitrospira or nitrosomonas, right? And then after we have nitrites, they're oxidized further into nitrates, and this is done by what? Nitrobacter.
Ah,
Lovely. All right. So what is D? We're changing nitrates now to atmospheric nitrogen.
denitrification.
Denitrification. What would be an example of an organism that does this?
Sir, please. My brain is screaming right now.
Pseudomonas is an example as well. All right. We also have Paracoccus or Pseudomonas, right, and some other bacillus species. They will assist. So Pseudomonas will assist here as well. All right. They do multiple things. Pseudomonas does a lot of stuff, right? And then we have E from the air from inorganic nitrogen to the plant directly. What would be doing that? Or what is that called first? What is that called? From atmospheric nitrogen to the air to the plant?
Assimilation, absorption.
No. I'll tell you why it's not. Right. This is nitrogen fixation.
Sorry, I just said that.
Yes, you did. Right. And this is done by the rhizobium. Right. Why is it that? Right. If it was the nitrogen, right, specifically, right, within the air, right, that's gonna be going towards the living plant, and that's nitrogen fixation specifically. If it goes directly to the plants, right? If it went to the soil first, right, then Azobacter and Clostridium could have helped to create it. Well, to turn it to ammonia, and then it would go into the plant. But generally, if we have something like that, we can actually have nitrogen fixation happening here. Directly from nitrogen in the air to the plant, that's going to be nitrogen fixation. And it's usually symbiotic fixation, which is through rhizobium. But Azobacter can actually fix them into nitrates and, well, not into nitrates. Azobacter can actually help with the production of them back into ammonia, so assimilation can occur. But it's directly from the air to the plant, generally fixation. Animals may also be involved in nitrogen cycle. Extend the diagram. So if we extend the diagram, how are the animals going to be looking now? So if we put an animal,
Let's say
well, go ahead.
I would say they will be eating the living plant. So I guess yeah.
And then
Consumption.
so
Decomposition.
so consumption and further decomposition. Lovely. All right. Energy is needed to metabolize nitrogenous compound within organisms. Oh, wow, never noticed. Under anaerobic conditions, right, the respiratory pathway differs depending on the organism. So take two similarities between fermentation reactions in animal and in yeast. There are two similarities. Both what?
So both of them only involve glycolysis.
Wait, what? Wait, glycolysis will occur before the fermentation, right? So both involve I guess glycolysis occurs before both. So glycolysis will occur before both of them, right? So glycolysis will always occur um before both of them, I guess. But what's another one? Any other one? Both of them use NAD, right?
Yes, sir.
Right. What will happen as well?
Both don't need oxygen.
Well, both don't need oxygen. Anything else?
And I think both produces a net amount of 2 ATP per glucose molecule.
2 ATP, right? Per glucose. Lovely. Right? 2 ATP is a lot for the yeast cell, but it's not a lot for the animal cell. Sadly, death will occur after fermentation occurs too long. Explain the function of fermentation reactions in the organisms. What is the function of the fermentation reaction? So what I'm gonna state first, right? Because I'm not sure if anybody would state it, but I'm pretty sure somebody would have. It's a primary. Go ahead.
To break down sugars.
Well, I would believe so. Yeah. Mm-hmm. So it's to break down sugar, but like that's not generally it though. Like that's the focus of all respiration, right? But what is the main function of fermentation specifically?
To continue the production of energy while oxygen is not present. Well, I mean, well, I guess. Oh yeah.
Sir, is not ATP and pyruvate.
Well, and its main function is it's going to produce pyruvate, but its main function is to try to produce ATP. Using it's gonna produce pyruvate now, but remember that pyruvate is gonna turn to lactate in animals, and the pyruvate is gonna turn to ethanol, right? Well, technically acetaldehyde and then ethanol. Well, acetaldehyde. Wait, is it acetaldehyde? And then ethanol in animals, right? So, well, in plants and back and yeast, I mean, right? For that. But the main process of fermentation is to continue to produce some amount of energy through ATP until oxygen depth can be replenished, right? So it will try and it will try and it will try to get some amount of energy without the use of oxygen until it cannot do it any longer. So it's basically a type of what is it called again? A backup
Yeah.
process. Go ahead.
Yeah.
It's a backup, right? But I also included this first line because people tend to forget it is the primary mechanism of respiration in some organisms. But for usually in multicellular organisms, right, they are a backup process, right? Usually, because plants don't really, well, plants generally use fermentation majority of the time. in the time of, well, it's really just plant respiration which produces ethanol technically. So it's not technically fermentation, but yeah. So we could just state that. So that's 3 marks though. That's fine. The process of oxidative phosphorylation is very important to the supply of respiratory energy under aerobic conditions. Discuss the importance of the following components of oxidative phosphorylation. What's the function of the proton gradient? Why do we establish proton gradients in bioenergetical in bioenergetic systems, especially when it comes to respiration? Why do we do it? I think somebody has to answer, but then want to give everybody else a try. But I would love it if the person just answer. What's the answer? Go ahead.
Sir, the importance of the formation of the proton gradient is to allow for these protons to move in such a way that they move through specialized carrier proteins known as ATP synthase and allow for the phosphorylation of ADP to ATP.
What is that process called?
Oxidative phosphorylation.
Not necessarily. Well, technically yes, right? But the establishment of the proton gradient, right, for that process, it starts
Is
with a
it
C.
electron?
Start with a C.
C.
Mm-hmm.
Chemiosmosis?
Chemiosmosis, lovely.
Yeah.
Right? So the main purpose is for chemiosmosis, and that chemiosmosis will create a flow of charged particles, technically a bioenergetical energetical system, right? That will now run through ATP synthase, right, and causes the complex to turn, and that turning mechanism, right? I think you guys have to do some biophysics to figure out how that turns, right? Where it's gonna create a turning mechanism that will combine an inorganic phosphate group to the ADP to produce ATP, right? So the purpose of the proton gradient is to establish, right? Well, the is to basically establish, right, some means for chemiosmosis to occur and the rapid production of ATP via ATP synthase. Lovely. The synthesis of ATP now. Why do we produce ATP? Go ahead.
Sir, because ATP is the unit of energy used in our bodies, so
Mm-hmm.
cells require ATP to produce energy to carry out their function.
Okay, so ATP is most definitely necessary. I think it's in the biology concise and Tyndale did state, right? Biology concise from CSEC that ATP is the currency of the cell. ATP is used in almost every single form where energy is needed. ATP is necessary for life as we know it, right? Well, multicellular life, anyways, right? So, ATP is necessary as it is generally the basic unit of energy within cellular systems. What is the role of oxygen? No. What is the role of oxygen in oxidative phosphorylation? And tell me where it is needed. Go ahead.
So I'm just gonna give somebody else a chance. Never mind.
Okay, go on. Anyone else? Where is oxygen needed? What is the role of oxygen? Come on, guys. This simple exam is tomorrow. Have a go at that. What's the role of oxygen?
All right, sir. Oxygen acts as the final acceptor in the electron transport chain and allows for the maintenance and continuation of the chain and the production of energy. It is found just at the base of cytochrome C oxidase, or yeah, I think so. The enzyme there.
So complex 4. Generally within enzymes in complex 4. And it is the final electron acceptor in the entirety of the respiratory process. The aerobic respiratory process. Right? So it is there to collect those final electrons that are passing through the electron transport chain and it produces water from that. Okay? So we have that there. And that is the end of the module 1 component. And we'll move into the module 2 component.